Hi everyone, my name is Caroline Maro. I'm a third-year PhD student at the School of Plant Science in the University of Arizona, and I'm working in the Anodes Lab and arid Center for my PhD project. Today, I am interested to talk to you about endophytes for the future, especially what Tepary shows us about surviving climate change. I know the term endophyte might be new to some of the people, so to define it, endophyte, these are microbes that are surviving inside the tissues of the living organism. So for plants, it's inside the plant tissues. It can be in different compartments like roots, seeds, leaves, flowers, etc. So bear that in mind as I take you through my talk. So let's see the impact of climate change to nutritional securities. Climate change, which encompasses rising in temperature, heat waves, and drought, threaten crop yield globally, and this causes declining in nutrient qualities of staple food, which threaten human health, especially in low-income regions. Research has shown that protein, iron, and zinc decrease dramatically under high elevated carbon and high temperatures. From the picture here, you can see the way the soil is very dry and also the very high temperature as a result of climate change. So this calls for a climate resilient, nutrient-dense crop that will overcome the impacts of climate change as well as dual challenges of hunger and malnutrition. One of the strategic solutions is consumption of leguminous crops, which are rich in protein, fiber, and key micronutrients. The highly consumed leguminous crop is common bean, especially in Latin America and Africa. However, this crop is highly sensitive to drought and high temperatures above 30 degrees, particularly during its flowering stage, which is the stage towards pod formation as well as yield. The combined stress of high temperature and drought has been proved to cause a yield loss of up to 60% to common bean. So this limited tolerance of this crop to these climate impact calls for an alternative regime that will better suit the future climate and secure food securities. And there is where Tepary is of great importance and of high importance to meet future climate impacts. Tepary bean as a crop is native to Sonoran Desert of southwestern North America. It is very nutritious, even more than common bean, and it thrives extreme temperatures and drought where other crops cannot survive. As you can see from the picture, we see a field which is really very dry, and this area is a very high temperature, but Tepary bean are very happily growing. And the key question is, what if the resilience of this crop is coming from something that is hidden inside its seeds? Particularly, what if it is in the endophytic microbes that survive inside the seeds of Tepary bean? So this was the foundation of my research interest, and I decided to research on the seed endophytic bacteria. To answer my basic questions, I isolated and I characterized at molecular level the seed endophytic bacteria of Tepary bean. I looked at their functional traits, looking at if they scavenge nutrients, if they have ability to tolerate very high temperatures, Tepary bean evolved from the desert area. How do they adhere and get into the seeds, as well as how do they promote plant growth? So to answer all these questions, I conducted also a field experiment to see the impact of seed inoculation with a single microbe and also the combination of these seed endophytic bacteria to the Tepary bean to see their impact in growth and yield of Tepary bean. And these characterization were coming from the high performing plants. My initial findings shows that there are four common endophytic bacteria that inhabit Tepary bean, as you can see from this first picture. Some of them, they look like alike in terms of their colony growth and color, but they are taxonomically and genomically diverse. These endophytic bacteria, they offer promising functional traits. For example, solubilizing phosphate and making phosphate available for plant. They can scavenge iron, so they help plant access iron, as well as they produce ammonia, which is the precursor for nitrogen, which is an essential nutrient for plant. And when I conducted a field experiment to assess the seed inoculation with these bacteria on growth and yield, I found out that these bacteria enhance both teperibine growth and the yield under arid condition, especially when they are in combination. So this picture here shows us some pods that were formed during this year's experiment, and when they dry, this is how they look like. So what I'm planning to do next is to explore the diversity of these seed endophytic bacteria across teperibine genotypes. So we have more than 200 teperibine genotypes, and I want to characterize the seed endophytic bacteria to look how diverse they are. I'm planning to evaluate the effect of these bacteria in nutritional qualities of teperibine. Are they contributing to the accumulation and concentration of nutrients in the seeds of teperibine? Also looking at the mode of action on the plant growth promotion, like which pathways they impact during plant growth, as well as looking the impact of these seed-borne bacteria to other regions or to other regions. Like, is the effect we see in teperibine same as in other regions such as common bean or any other region? Lastly, I would like to acknowledge my supervisors, Dr. Betts Arnold and Duke Paulai, for their support during conducting of this experiment and my research, members of Arnold Lab for their help during conducting of my research, isolation and everything, and lastly, the sources of funds that enables this project to be carried through. These are the references that I used for my work, and I'm glad to take any question. Thank you so much.